Thanks, Tim, and welcome everybody to the session today. The development of stem cells represents one of the major new waves in medicine, and quite simply, stem cells are the body's own mechanisms for repair, recovery, and regeneration. And the ability to harness that allows us to approach the treatment of disease and recovery of diseases in a number of very, very important ways. The particular type of stem cell that is receiving most attention at the moment is a mesenchymal stem cell, or MSC. And these cells represent a promising treatment for a broad range of diseases. And what we're now seeing is that clinical success in a range of clinical trials is creating a very large demand for product. And because these are living cells or living drugs, the manufacture of such products is very complicated. So ultimately, commercial success requires a viable, scalable manufacturing solution that ensures a very consistent product. And we've just seen recently some very informative news from the FDA about that particular issue. And the FDA's reminded manufacturers in this area to ensure that their products are consistent. And that is where Cynata's Cymmerus platform technology for the manufacture of MSCs provides a very, very important solution. So moving to the next slide, I'll explain a little bit about how our technology works and how it differs from the current approach or the conventional process to manufacturing MSC-based therapeutics. The conventional process is indicated there at the top of the slide, and it involves accessing cells from donors, human donors who are prepared to donate mostly bone marrow or fat tissue, from which the product, the MSC product, is purified and isolated. The product is then expanded in tissue culture when sufficient quantities are made, so cells divide 2 to 4 to 8 to 16 and so on, and when sufficient quantities are obtained through that manufacturing process, they're isolated, and then the product is derived and used in the patient. This process, as I mentioned, requires multiple donors. And of course, since every donor is different, we're all individuals, there is no ability to ensure consistency because everybody has different MSCs, and the properties of those MSCs have been shown to differ from different donors, a point that the FDA, as I said, has recently expanded upon. The Cynata process, which is covered by our patents, involves a completely different transformative approach where we use cells derived from a single donor in a one-time donation to derive what's called an induced pluripotent stem cell, or an iPSC, from which our finished product can be manufactured, ensuring, as we only use one donor, that we have a highly consistent, reproducible, and very potent product. And ultimately, this process assures regulatory agencies around the world of consistency and quality, and ultimately effectiveness in clinical trials. And that's borne out too in potency, where our product requires typically only about 2 doses per patient versus much higher doses, which in the bone marrow derivation or adipose tissue derivation strategies, which of course is a huge cost of goods advantage and practical advantage for our technology. If we move to the next slide, we have now translated this manufacturing technology into clinical trials and clinical development. And of course, ultimately demonstrating that your product is effective in the clinic, in the disease targets in which you're pursuing, is the key to commercial success. Thus far, we've completed a very successful clinical trial in graft-versus-host disease, a condition that arises in cancer patients who have had a bone marrow transplant. And that technology, or that product, has now been licensed globally to a major pharmaceutical company, Fujifilm, who are now pursuing further development of that product at their cost. We're moving forward with a phase 3 clinical trial in osteoarthritis, a program in COVID19, which I'll talk more about in a moment, critical limb ischemia, for which we have approval, and multiple other preclinical development pathways, which we can then produce or move forward into clinical trial very soon. Moving to the next slide, To show where the valuation opportunity lies in an investment in Cynata, I think it's important to give you some indication of comparables. I've got a couple of companies here who are developing cell therapy products based on the iPSC strategy. One, BlueRock, was acquired recently, or a year or so ago, in a major acquisition. Previously, it was a private company acquired for $1 billion. Fate Therapeutics, a US-listed cell therapy company, also using iPSCs as their manufacturing strategy. And an Australian company that many investors will be familiar with, Mesoblast, which is developing mesenchymal stem cells. Their market cap now is over $2 billion US. They have multiple targets, but they're relying on the bone marrow derivation strategy that I mentioned before. So if you're looking at where upside opportunities lie, it's very clear here from these other comparable companies that there's a heck of a lot of blue sky ahead for Cynata, who is now in multiple late-stage clinical trial with a very, very attractive manufacturing solution. If we move to the next slide, ultimately the driver for value for biotech companies is clinical data. To be able to show that the company's product is effective in particular diseases. And in our case, we've conducted a clinical trial in graft-versus-host disease, which is a very severe condition arising in bone marrow transplant patients, wherein we demonstrated outstanding efficacy. These patients essentially have no option. They have failed the only course of treatment available to them, which is corticosteroids. And in that clinical trial, on the left-hand panel there, you can see that we achieved all efficacy endpoints. The complete response in these patients 100 days after they received the product of 53%, and a survival rate at that time point of greater than 87%. The high response rates that we've demonstrated there would be consistent with an approvable product should we move forward to phase 3. And of course, that's now in the hands of our partner, Fujifilm. On the right-hand side of the slide, you'll see some recently released data from a 2-year follow-up of these patients, wherein we assessed survival 2 years after the completion of the study. And we were delighted to see that we had a 60% survival rate. Now that may not seem great, but bear in mind these patients have a very grim outlook. And on the lower right-hand side there, you'll see that typically with standard of care, we would expect to see only around 17% survival. And compared with other MSC products, our 60% survival is also very, very interesting because it's much higher than those other products have reported. Most importantly as well, we saw no safety signals and no adverse events. Moving to the next slide, see how we intend to build shareholder value. And ultimately continuing to drive forward clinical development and generate clinical data to demonstrate safety and efficacy is, of course, the main pathway for biotech and pharmaceutical companies. But Cynata also has a license-driven or partner-driven strategy, which we are very aggressively pursuing, and our deal with Fujifilm is one such example of success in that area. And ultimately also strategic exits or mergers could also be a way to build and derive shareholder value. And in fact, last year, Cynata was the subject of a takeover bid mounted by Sumitomo Dainippon, Japan, at $2 per share. We decided not to proceed with that at the time for a number of reasons, but that leaves the company still in a great place with our deal with Fujifilm, as you can see on the right-hand side there, producing, if ultimately successful, a very substantial revenue flow to the company, in addition to the $4 million Australian in equity that we've already received at a 35% premium, and a nearly $3 million milestone payment that we received late last year when the deal was signed. Most importantly too, such a transaction validates the company's technology. It shows that a global pharmaceutical company, having done its due diligence and completed its review of the technology, has decided that it's worthy of investment. And that is certainly a very, very useful data point for investors in this complicated space. Moving on to the next slide. I mentioned briefly that we have a phase 3 in osteoarthritis soon to get underway. This is a very large clinical trial in patients with osteoarthritis. 440 patients will be randomized one-on-one to receive either placebo or our product. So it's a very well-designed trial and with the power to demonstrate whether or not MSCs ultimately are effective in treating this particularly challenging condition. As many of you will be aware, osteoarthritis is a progressive disease that really can only be controlled by means of pain relief and then ultimately surgery. So using a product such as an MSC to intervene and potentially reverse the course of this disease would be a fantastic outcome. It's now been approved by the ethics committees, and we await the final announcement by the state government of New South Wales that the restrictions around trial recruitment have been lifted. This not only affects Cynata, but many other companies. It's a very large commercial opportunity, over $11 billion, as most investors will be aware. Next slide, a very topical program at the moment is in COVID-19. MSCs have been shown to be potentially useful in treating patients with severe complications arising from infection with SARS-CoV-2, and this trial will be in patients who are admitted to intensive care wards with compromised lung function. It builds on some very exciting preclinical data we've already demonstrated in 3 different areas that are highly relevant to these patients. And it's important to note that in these patients, it's not so much the infection itself that's causing the problems, it's the body's reaction, the immune reaction to the infection. So bringing that immune response under control is the key to, we believe, to success in getting these patients out of ICUs and back into recovery. It's a 24-patient clinical study, and recruitment is now open for that trial in New South Wales. Moving to the next slide, the wrap-up. So in summary, Cynata's technology is a scalable, globally applicable technology for the manufacture of this very exciting type of stem cell, an MSC. We've got ticks in the major boxes of importance to investors. Successful clinical trial results where we have shown the product works in patients with a disease as opposed to healthy volunteers. We've got a clear pipeline of high potential target areas, so multiple shots on goal, and an attractive licensing driven business model, which we have already executed on in our partner deal with Fujifilm. And as the business of regenerative medicine continues to grow, Cynata is extremely well placed to capitalise on that with our technology. So thanks, Tim. Open for questions.
Thanks, Ross. Just a couple of questions. We look like we've got a couple of people who know what they're talking about in this space. So Mesoblast has kind of a lock on the IP in this space. Is that correct? And kind of how do you move around that?
Well, it's not for me to comment on Mesoblast's IP, but Mesoblast was one of the first movers in this area, and their technology, their intellectual property, is mostly around the process for purifying and isolating MSCs from bone marrow. Our technology takes a completely different approach to that. We don't require isolating any cells from anybody's bone marrow, and so therefore we have a completely different strategy.
And just the kind of the partnerships, is that the way you move forward for distribution, and do you kind of lose margin as you do that moving forward?
I think, as Bianca pointed out in her opening remarks, building a fully integrated pharmaceutical company requires an enormous amount of capital, and that includes getting all the way to market, so building sales and marketing resource in major markets around the world. So ultimately, a company in the Australian context is better served by accessing the capital, the resources, the expertise, and the global outreach that comes with established pharmaceutical companies. And that's why we did a deal with Fujifilm. Yes, we do leave money on the table because obviously we're not capturing full value of the technology as if we were selling it ourselves. But on the other hand, we spread the risk, and of course, we considerably spread the cost.
Yeah, understood, Ross. One last question. So what's the main difference to your cells and those of the many other companies in the space?
Well, that's a very good question. Mesenchymal stem cells are broadly defined by a number of characteristics, but there clearly have been established major differences between MSCs that are isolated from different individuals, that are cultured over various periods of time, and various other means in which an MSC can be defined. Ultimately, whether those changes in characteristics translate to differences in clinical outcome in patients in whom they're used is an open question. What we know about our product is that it is very, very effective and very potent. And I think that's the most important thing for Cynata and our investors.